I will say that AI in drug discovery is a very different application just because, as you mentioned, drug discovery really has big data. And so AI can do an incredible job at sifting through millions of data points and finding trends that's really hard for humans to identify. In some ways, it's the perfect fit, which is why I think we're seeing so many companies from massive biopharmas to new startup using AI in the drug discovery space.
Hey there, Smart Biotech Scientist. This is David Brühlmann, your host, and today we are diving deep into the future of bioprocessing with a guest who is bringing Wall Street's quantitative power to our bioreactors. Meet Yossi Quint, who is the founder and CEO of Ark, who made the fascinating leap from quantitative trading at 2 Sigma to revolutionizing pharmaceutical manufacturing. Get ready to discover how computational tools and AI are transforming the way we develop and scale bioprocesses. Stay tuned for part 1 of this mind-expanding conversation. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist Podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Yossi, it's great to have you on today.
Thank you so much, David. I'm a long-time listener, and so it's a real honor to join you today, too.
I'm excited about today's conversation. So, Yossi, since you've listened to so many episodes already, I am curious. So share something that you believe about bioprocess development that most people disagree with.
Within the next decade, the most innovative companies are going to replace over 90% of their bioreactor experiments with simulation. And they're going to run hundreds of simulations in place of each bioreactor run. And that changes everything. Imagine if you can run 1,000 more experiments, and running all of those experiments takes less time than running just a single physical bioreactor in the lab. Experiments are the lifeblood of science, and more experiments ultimately means better outcomes. Simulation, computational bioprocess, using models means that you can have an entire team of scientists working for you. And they work in hyperspeed. And so by running more experiments in silico, scientists will need to run fewer physical experiments while still getting great answers. In fact, they're going to get even better answers, which means that they're going to have more time to run more molecules. If you could reduce experiments by 90%, that means that you could 10x throughput. Now imagine what happens when we take 10 times as many shots on goal. How many more life-saving drugs can we bring to market?
Yeah, definitely. The future is bright. If we could reduce our experiments by 90%, that will be a game changer. Before we dive a bit more about how are we going to achieve that, I would love to hear from you, Yossi, because your journey from trading to biotech is fascinating. So draw us into your story. What sparked that transition and what were some interesting pit stops along the way?
My first internship ever was on the floor of the New York Stock Exchange, the heart of Wall Street. And this was back when traders shouted at each other, there were paper tickets littering the floor, and decisions were based on individuals' expertise. Each trader relied on their gut instincts and just that muscle memory that they built up over decades. And my job back then as a lonely intern was typing those paper orders, those paper tickets into the computer, bridging the analog world with the digital world. After I graduated college, I went in a similar but different direction. I joined a quantitative hedge fund. And the contrast was stark. No shouting, no papers littering the floors, just algorithms executing trades. And that those algorithms were built on the collective intelligence of hundreds of people encoded in software code. And so instead of having a few experts making the best decisions that they could make based on their own personal experience, you had this collective wisdom being made automatically, algorithmically. I then took a bit of a different direction. I went to join McKinsey, a management consulting company, where I had the opportunity to work on digitization projects and work with a bioprocess company. And I saw that this company was doing some of the most innovative work in the entire world in the lab. But bringing that work from the lab to the masses, that bioprocess step, was really challenging for them. And it wasn't just challenging, it was super time consuming. They had a room full of high throughput bioreactors running 24/7. They were spending tons of money, and yet they weren't getting the results they wanted. And coming from this quantitative hedge fund world, I saw just how much you could do with algorithms and just how precious every piece of data was. And there I was working with this bioprocess company, and I didn't feel like they were taking full advantage of their data. And I'm a very mission oriented person. And I really care that we're bringing great science to the masses, especially in the case of pharma, where we're saving people's lives. And I just thought there was so much opportunity to bring better quantitative tools to empower scientists. So I quit my job. And after spending a bunch of months trying to find the right technical co founder, I started Ark. And what we're doing at Ark is we're trying to bring that same quantitative approach to the bioprocess space through capturing decades of scientific knowledge and packaging that into software using the best of scientific mechanisms along with the best of machine learning and AI. We're packaging that into really easy to use software so that scientists can have the power of quantitative tools and make better decisions.
Since you have seen two completely different worlds, the trading and now biotech, are there some unexpected lessons from your hedge fund days that you're now applying in biotech?
Yeah, that's a great question. Certainly. So, one of the things that I saw in the hedge fund world is just how much we could do with data and how much we could bring that scientific process that scientists are so familiar with and bring that into a data driven space. And what I saw in the hedge fund is that through being really thoughtful around how do we train algorithms, how do we test algorithms, we can deliver algorithms that have tremendous predictive capability. Ultimately, what we're talking about is how can we create high predictive simulations? And quant finance did an incredible job leveraging simulations to predict performance. Through running thousands of experiments virtually before making a physical trade, they could yield better outcomes. And that's the exact same thing that we're working on in bioprocess of how can we run thousands of experiments before we run a physical experiment so that each experiment has the highest impact.
Now, we have seen tremendous innovation in bioprocessing. We have new types of bioreactors. We have single bioreactor approaches. We have small scale, mini scale. We have models, high throughput systems, you name it. What key challenges has each wave addressed, and how are you now solving the challenges we are still facing in bioprocessing?
One of the great things about scientists is that they are so innovative and so curious and care deeply about bringing the best technology to their work. And so, to me, looking at the history of bioprocess is a really great example of scientists continuing to innovate and continuing to bring that best technology. You could start off thousands of years ago with the fermentation of food products, but to make it a little bit more modern, let's start off in 1945, where we had the first industrial bioreactor that was used to make penicillin. And the goal of that first bioreactor was how can we create enough product to actually have impact on the masses? So how can we go from the lab to making something that will help people in the real world? You then have to fast forward a few decades to the next great bioreactor innovation, which was in the early 2000s, single use. And single use was about how can I reduce the time between batches and reduce the risk of contamination. Fast forward a few more years, and the automation project introduced the AMBER system, the first high throughput bioreactor. And AMBER stands for the automated microbioreactor. How can we automate, bring more efficiency, and move from running bioreactors sequentially to in parallel? How can we run 24 bioreactors at the same time with a similar effort it would take to run one bioreactor? And high throughput is incredible, which is why it's become the status quo. And that's because experiments are the lifeblood of science. Experiments are how we learn, how we get better. The next wave of bioprocess is going to be computational. It's going to be truly high throughput. How can we go from running 24 bioreactors in 2 weeks to running 24 bioreactors in 2 minutes? Imagine what's possible. Instead of running 24 experiments in a month, we run 24,000 experiments. That's a lot more learning.
Yeah, absolutely. This will be a total game changer. Now, for those of the scientists listening today who are not familiar with, I'd say, the classical development workflow, can you break it down? How does a typical bioprocess development timeline look like today? And where ideally are we going to take this tomorrow?
Even before you start an experiment, you have a hypothesis that you want to test. And then you design the experiment to test that hypothesis in a rigorous way. Often scientists will use statistical tools like JUMP. Then you start to set up the bioreactor. That could take hours, it could take days if it's a really big system. And then you start the experiment. And the experimental timelines are limited by how fast the cells can grow. You can't get the cells to grow faster. And so a typical experiment might be a week or 2 weeks. If you're doing a perfusion process or continuous process, it could be weeks to over a month. And during that entire experiment, a scientist is going to be coming in, they're going to be collecting data, taking samples, they might be preparing media. And so you're pretty busy. Once that experiment ends, you then might have to send the results to an analytics team who'll do additional work, and then you're going to get back all that data. You're going to clean that data, do data analysis, and see whether your hypothesis was proven or if you need additional experiments and you need to repeat this process all over again. So the feedback loop is really, even the fastest best-in-class feedback loop is still limited by how fast cells can grow. And so the beauty of computational bioprocess, of moving experiments in silico using simulation, you could speed up that step of the cells growing. Instead of having to wait weeks for cells to grow, you could wait seconds.
Now let's unpack this, the in silico models and how you do that, because we have different technologies. We hear about hybrid modeling. We hear about large language models, AI. We have now DeepSeek, we have the new models and taking over. There's so much going on. It's just very hard just to stay on top of all the news. What is your approach? How do you leverage in silico models and how do you train these models, or what's the basis of these models?
As you mentioned, there are a lot of different approaches. And I would say that if you wanted to really break this down, there's primarily three big approaches. The first one is a purely data-driven approach. So I'm taking the data, and based on that, those observations of the data, I'm doing some sort of computational work. This is going to be purely statistical. It could be using very traditional statistics like linear regressions, or it could be using more advanced tools like machine learning or AI. The second approach is a mechanistic approach, which is about using scientific models. So really understanding what's happening at the scientific level and modeling that. This is sometimes called mechanistic modeling or multi-physics modeling. And this is about combining the best of biology, chemistry, and physics. The third major bucket is hybrid models, as you mentioned. And hybrid models are about combining these mechanistic scientific models with these data-driven models. And within that, there's a lot of different gradations because it could be mostly data-driven or mostly mechanistic. And so it's a big bucket. At Ark, we use a hybrid modeling approach. At the core, a mechanistic approach is the gold standard because if you truly understand what's going on in the system, that's perfect, that's ideal. But in reality, our knowledge is limited. We just haven't gotten to the depths of knowing exactly what's happening, especially on a cellular level, just because bioprocess is so complicated. And so the benefit of bringing in some of these data-driven approaches to complement the mechanistic model is that we're able to build a comprehensive picture of the system. And so there's a real opportunity for some of these statistical approaches, the newest approaches that are leveraging AI to complement the mechanistic approaches and to enable us to have predictive power where we don't yet have a mechanistic understanding of the system. I like to think about this as a boxer going into the ring. It's great to go into the ring and be going at it with one hand, but you don't want to be boxing with one hand tied behind your back. If you're just using a mechanistic approach, using a data-driven approach, you're leaving opportunity on the table.
And how does this practically work, Yossi? Let's assume I'm leading a startup, I'm the CEO of a startup, and I have this new life-saving therapy, and we're developing a process. How much time would you need to train the model and to then come to a place where you could cut 90% of the experiments? How long would that take, and what would be 2, 3 major phases to get there?
That's a great question. So at the core, because the models have somewhat of a data-driven computational component, we need to leverage some historical data. Depending on what the application is, the number of historical runs will vary. As a rule of thumb, we like to have around 10 runs of historical data where there is variance in the data. So if you've just run 3 sets of triplicates, you're not going to really be able to train the model in as comprehensive a way as if those 9 runs are actually exploring different parts of the design space. So let's say a high pH or lower pH, a higher temperature or lower temperature. Once we have that data and we calibrate our model, you're then able to use it across scales, across modes of operation. So you could use it to simulate a perfusion process, or you could use it to simulate a fed batch process. You could simulate a 2 liters, you could simulate a 20,000 liters. And so we don't need data from different modes of operation, from different scales. We just need data at one scale or even multiple scales where there is a diversity of conditions being tested.
But that means that we need to start right, which means in the beginning, it's extremely important to have a data set with a lot of variability, right?
There's tremendous benefit of having variability in the initial data, yes.
And to what extent can now AI help us in this process? Because obviously there's a lot of hype, but AI is also very powerful. But we still have this limitation in bioprocessing because we don't have these huge data sets. So there is some limitations and there's some trade-offs. But to what extent do you see AI facilitating this whole process?
So AI has a number of roles it can play. I would bucket AI into 2 larger types of opportunities for bioprocess. The first one is supporting activities, and the other one is core activities. The supporting activities are the lower hanging fruit. Those are opportunities that scientists can use today without too much activation energy. And that's things like automating data cleaning, finding relevant academic literature, or optimizing workflows. These are things that traditional AI models that are off the shelf are already really good at. The second opportunity is actually bringing AI into the modeling effort. And this is where things get really exciting. In my view, AI has a meaningful role to play in these algorithm development, especially when it comes to modeling cell metabolism, where the mechanistic models often fall short, at least today. Metabolism is extremely complex, and existing mechanistic models struggle to fully capture it. And those models that try to capture it often take weeks to run, which is often just not useful in an industry setting. And so, to me, a really great use of some of the AI computational approaches is for these metabolic models.
Well, we hear a lot about, research and development or discovery companies. And since they have a lot of targets, a lot of hits, they have huge data sets, and AI definitely is a game changer there. How do you see this changing as we are moving into bioprocessing? Will there always be like an end approach, hybrid models and other more traditional combined with AI, or where is the limitation actually? Where's the ceiling?
I definitely don't have a crystal ball. And given the incredible leaps and bounds that AI is taking, what seems like every few days, who knows where we're going to be in a month or two, let alone a year or two. I will say that AI in drug discovery is a very different application just because, as you mentioned, drug discovery really has big data. And so AI can do an incredible job at sifting through millions of data points and finding trends that's really hard for humans to identify. In some ways, it's the perfect fit, which is why I think we're seeing so many companies from massive biopharmas to new startup using AI in the drug discovery space. Now, I do think this will have a knock-on effect in bioprocess because if you're having more molecules discovered faster, it means that bioprocess teams are going to have to do more work. they're going to have more molecules to be tested to get to clinic. And we're also seeing AI being used a lot in clinical trials on the other end of the spectrum. And so I do think that Bioprocess is going to want to really stay on top of trends and to continue to bring AI in. The other thing that I'll mention is that Bioprocess is doing a really good job in digitization. And so every major company has a digitization effort underway where they're working to create better data lakes, better storage, thinking about how to include metadata. And so as that becomes more mature, which is an active process, we're going to also be able to use more of these big models to query historical data and use them in day-to-day decision making.
And that wraps up part 1 of our conversation with Yossi Quint. Fascinating how computational approaches are reshaping bioprocess development, right? Join us next time for part 2, where we will explore more about in silico modeling and its impact on tech transfer. Before we go, if you are wrestling with process development challenges, I'd love to help. Book a free process development assessment call with me at brühlmannconsulting.com. See you in part 2. All right, smart scientists. That's all for today on the Smart Biotech Scientist Podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.